Hi, welcome back to my class. My name is Satwika, and in this video, we'll be continuing our discussion about the development of our brain. All right, so before we move on, think all of the things that you can do right now, but you couldn't have done a few years ago. So for example, like right now, you can analyze statistics, you can read a foreign language, or maybe you can write brilliant critiques of complex issue and so on. The question is, have you developed these new skills because of brain growth? Well, actually, many of our dendrites have grown new branches, right? Because we're learning new skills. But your brain as whole hasn't grown at all. But you may also think about things that 1-year-old children can do that they could not do at birth. The same question, have they developed their new skills because of brain growth? Well, the answer to large extent, yes, their brain grew, although this result depends on experiences as well. So that's why in this module, in this chapter, we consider how neurons develop and how their axons connect and how experience can modify its development. All right, before we moving on forward, we should discuss about the maturation of the vertebrate brain first. Is the homeobox genes. There's something that called homeobox genes. So the earliest stages of development are remarkably similar across species, yeah, actually. And a series of genes that known as homeobox genes could be found in all species, including vertebrates, insect, plants, even fungi and yeast. And these homeobox genes regulate the expression of other genes and control the start of anatomical development, including such matter as which end is the front and which is the rear, right? And all these genes share large sequences of DNA bases, and a mutation in one homeobox genes can cause mutation, right, or disorders. So in homeobox genes in humans have been linked to many brain disorders, including mental retardation as well as physical deformities later in life. So we should recall that the human central nervous system begins to form when the embryo or the fetus is about 2 weeks old. So you can imagine that. So it is very important to be mindfully healthy in the first semester of pregnancy, because it is very critical for the infant, of the brain development of the infant. So first of all, the dorsal surface of the, what we call in the future will be a brain, will thickens, and then long thin lips rise, curl, merge, and then forming a neural tube that surrounds a fluid-filled cavity, as you can see in this figure. And then after that, as the tube sinks under the surface of the skin, the forward and enlarge and differentiate into the hindbrain, midbrain, and forebrain, as you can see in this figure. Yeah, they start to develop into the anatomy of human brain. And then also the rest become the spinal cord, yeah, and will become the back of a human being. And the fluid-filled cavity within the neural tube becomes the central canal of the spinal cord and the 4 ventricles of the brain, as you can recall from the previous classes. And inside the ventricles, then it's also developing a cerebrospinal fluid. And then also the first muscle movement will be started the 7th weeks, yeah. And their only accomplishment is to stretch the muscle. And that's why at that age, then spontaneous activity in the spinal cord drive all the muscle movement. So as the sensory organs are not yet developed or not yet functional, but the muscle movement started at that age. That is contrary to what we might guess, that we start making movement before we start receiving sensation. All right, so in early infancy, after the human was born, the average human brain weighs about 350 grams. And then by the end of the first semester, it weighs 1 kilogram. Yeah, so it's really close to adult brain weight, around 1.2 to 1.4 kilograms, right? So it means that by the first year of life, then human beings will rapidly grow their brain. And then also brain maturation in early infancy, the primary sensory areas of the cortex responsible for registering vision, hearing, and other senses are more mature than the rest of the cortex. Their gyri and sulci are mostly formed already, and their connection with the thalamus are fairly well established. They continue to develop, of course, but the greatest changes over the first couple of years happen in the prefrontal cortex and other cortical areas responsible for attention, working memory, and decision making. So in short, the infant brain is set up to see, hear, and so forth at first, right? But then limited in its ability to interpret that information or to decide what to do about it. So the human prefrontal cortex then continues slowly maturing through the teenage years and beyond. So in general, the brain areas that are slowest to develop, such as the prefrontal cortex, are the one most likely to deteriorate in conditions such as Alzheimer's disease. And neuroscientists here distinguish the several stages of the development of our neurons. Yeah, there are proliferation, migration, differentiation, myelination, and synaptogenesis. Let's check them more closely. So the stage one is the proliferation, is the production of new cells. So early in development, yeah, the cells lining the ventricles of the brain divide. Okay, some cells remain where they are as stem cell, continuing to divide, whereas other migrates to other parts of the nervous system. All right, so in the stage of proliferation, most of this stage then later will continue to the migration stage later, and it all occurs before birth, okay? But a small number continue to migrate for the first few months after birth, okay? And one of the major differences between human brains and chimpanzee's brain is that human neurons continue proliferating longer than the chimpanzee brains. Okay? And nearly all neurons form within the first 28 weeks of gestation, and that's why like premature birth before that time will inhibit slow neuron formation. And we're moving on to stage two. So in the migration state, early in development, the primitive cell is not yet identifiable as neuron or glia. And at that stage, they begin to migrate or moving to other part of the body. So chemicals known as immunoglobulins and chemokines is responsible in guiding the migration of our neurons. All right, so immunoglobulins and chemokines is some sort of a protein that is important for guiding this migration stage. Those a deficit in these chemicals will lead to impaired migration. Yeah, it can cause the decrease of brain size, and like also that will cause mental retardation later when the baby born. And as well, the brain has many kinds of immunoglobulins and chemokines here, reflecting the complexity of brain development. And after the migration, continuing to the stage of differentiation, so as a cell differentiates into a neuron, it begins to form its dendrites, axon, and synapses. Okay, and the next stage is the synaptogenesis, is the formation of synapses. Yeah, remember what is synapses? So it's the gap between the neurons. So the synapses begin long before the birth, yeah, but it continues throughout life as neurons form new synapses and discard old ones. And the process is generally slow in older people, as does the formation of new dendritic brain cells. And later, and slower stage of neuron development is the myelination. It's the process by which glia produce the insulating fatty acids that you can recall from our previous classes. That is accelerate transmission in many vertebrate axon, right? The myelin seed. So the myelin formed first in the spinal cord, then in the hindbrain, midbrain, and forebrain. Okay, and then this myelination continues gradually for decades and increases as a result of learning a new motor skill. Okay, so the question is, can the adult vertebrate brain generate new neurons? According to the traditional belief dating back to Cagel's work back um, in 1800 something, is that the vertebrate brain form all the neurons in embryological development or early infancy at the latest, and afterwards they are not growing anymore. But beyond that point, neurons could modify their shape, right? But the brain could not develop new neurons. But later researchers found exception. So the first exception were the olfactory receptors, which are exposed to the outside world and its toxic chemical. So the nose contains stem cell, yeah, that remain immature throughout life, and periodically they divide with one cell remaining immature while the other differentiate to replace a dying olfactory receptor. It grows its axon back to appropriate site in the brain. So the olfactory receptor send axon to the olfactory bulb, and later researchers also demonstrate that the formation of new neuron in the olfactory bulb for many species as well. However, new neurons do not form in the area for humans, at least after the first year or so of life. And another example is in songbirds. One brain area that is necessary for singing loses neuron in fall and winter, and they regains them in the next spring where the mating season happening, and they will be able to sing again. Also, new neurons form in the adult hippocampus of birds and mammals. Found out that that the hippocampus is important area for memory formation, right? And a supply of new neuron keeps the hippocampus young for learning new tasks. And other findings suggest that blocking the formation of new neurons, such as by exposing the hippocampus to X-ray, yeah, will impair the formation of new memory. All right, and then let's continue to the question of how do the developing nervous system find their way. So I imagine this: if you ask someone to run a cable from one place to another place in your room, your direction could be simple, right? Just go straight and then found the cable, for example. But imagine asking someone to run a cable to somewhere on the other side of the country. Yeah, you would have to give detailed instruction about how to find that right city, the right building, the location building, within the building. So actually the developing nervous system faces a similar challenge like that, because it sends axon over great distance. Yeah, and the question is, how do they find their way? A famous biologist called Paul Weiss in 1924 conducted an experiment in which he grafted an extra leg to a salamander and then waited for axon to grow into it. So unlike mammals, salamanders, and other amphibians accept transplant of extra limbs, and they can generate new axon branches to the extra limbs, right? And much research requires finding the right species to study. And after the axon reaches the muscle, the extra leg move in synchrony with the normal leg next to it. So based on that experiment, Weiss then dismissed the idea that each axon found its way to the correct muscle in the extra limb. He suggested that instead that the nerve attached to muscle at random and they send a variety of message, each one tuned to a different muscle. He supposed that muscles were like radio tuned to different station. So each muscle received many signals but responded to only one. But later evidence supported the interpretation that that Weiss had rejected. So actually, using the salamander again, the salamander extra leg moved in synchrony with its neighbor because each axon found the correct muscle. So continuing the Weiss experiment, Roger Sperry, he is a former student of Weiss, performed a classic experiment that showed how sensory axons find their way to their correct targets. The principle is the same as for the axon finding their way to the muscles. What Sperry did is, firstly, Sperry cut the optic nerves of some newt. Note that the importance of choosing the right species, right? A cut optic nerve grows back in amphibians, but not in mammals or birds. So the damaged optic nerve grow back and connected with the tectum, which is amphibian's main visual area, you can see in this figure, thereby re-establishing normal vision. So then Sperry's question was, did they grow at random or did they grow to a specific target? And then continuing his experiment, Sperry then cut the optic nerve and rotate the eye of the newt by 180 degrees. So when the axon grow back to the tectum, the axon from what had originally been the dorsal portion of the retina, which was now ventral because it's rotated, grow back to the area responsible for vision in the dorsal retina. So axon from other parts of the retina also grow back to their original targets. So the new newt now saw the world upside down and backward, responding to stimuli in the sky as if they were on the ground, and to stimuli on the left as if they were on the right. Okay, so each axon actually regenerates to the same place where it had originally been, presumably by following a chemical trail. It's really interesting, right? So the next question then, how does an axon find its target? So the current estimate is that humans have fewer than 30,000 genes in total, right? If you can recall from the genetic class, and probably fewer than 20,000, far too few to specify individual targets for each of the brain billions of neurons. And then a growing axon follows a path of cell surface molecule attracted by certain chemicals and repelled by others in a process that steer the axon in the correct direction. All right, so eventually axons sort themselves over the surface of their target area by following a gradient of chemical. So by analogy, you could think of men lining up from tallest to shortest, pairing up with women who line up from the tallest to shortest. So it's how they can find the path of the axon by chemical gradient. All right, so continuing to the competition among axon as a general principle. So when axon initially reach their target, so the chemical gradient steer them to approximately their correct location, but it would be hard to imagine that they achieve perfect accuracy. Instead, each axon forms synapses onto many cells in approximately the correct location, and each target cell receives synapses from many axons. So over time, each postsynaptic cell strengthens the most appropriate synapses and eliminates other. And this adjustment depends on the pattern of input by incoming axon. So for example, here, one part of the thalamus receives input from many retinal axons. And during the embryological development, long before the first exposure to light, repeated wave of spontaneous activity sweep over the retina from one side to the other. And consequently, axon from adjacent area of the retina then send almost simultaneous message to the thalamus. And each thalamic neuron select a group of axons that are simultaneously active. In this way, it finds a receptor from adjacent neuron of the retina. It then rejects synapses from other selection. Sorry, from other location. All right. So actually, like getting the right number of neurons for each area of the nervous system is more complicated than it might seem. Consider an example. So like the sympathetic nervous system send axon to muscle and gland. Yeah, so each ganglion has enough axon to supply the muscle and gland its area with no axon left over. So the question is, how does the match come out so exact? So one hypothesis was that the muscle send chemical message to tell the sympathetic ganglion of how many neurons to form. And researchers named Levy-Montalcini in 1987 was largely responsible for confirming this hypothesis actually. So she discovered that the muscle do not determine how many axons form, but they determine how many survive. So initially the sympathetic nervous system forms far more neuron than it needs, and then when one of its neuron form a synapse onto a muscle, that the muscle deliver a protein called nerve growth factor, or NFG, that promotes the survival and growth of the axon. And then an axon that does not receive NGF degenerates, and the cell body dies. Right, that is, each neuron starts life with the suicide program. So if its axon does not make contact with an appropriate postsynaptic cell by a certain age, the neuron will kill itself through a process called apoptosis. That is a program mechanism of cell death. Okay, and then this NGF cancel the program for apoptosis actually. It is the postsynaptic cell's way of telling the incoming axon that I'll be your partner, don't kill yourself. And then the sympathetic nervous system way of overproducing neuron, and then applying apoptosis enables the CNS to match the number of axon to the number of receiving cell. Okay. And nerve growth factor is actually is neurotropin. It's meaning a chemical that promotes the survival and activity of neurons. In addition to NGF, the nervous system responds to brain-derived neurotropic factor, or BDNF. That's also important to be remembered, and several other neurotropins. All right, and according to now we're moving to the vulnerable of developing brain, and according to Louis Wolpert in 1991, he said that that gastrulation is the most important time of our life. Yeah, so gastrulation is one of the early stage of embryological development. So Wolpert's point was that you mess up in early development of the brain during the infancy, you have problem from then on. Okay, actually, if you mess up during gastrulation, your life is over because it's affecting the development of neural system. So during early development, the brain is highly vulnerable to malnutrition, to toxic chemical and infection that will produce milder problem at later age. Okay, so for example, when we have a impaired thyroid function when we are adult, will only cause a lethargy. But if it happens during the infancy, then it will cause mental retardation, right? Another example is as an adult, we have a fever. It may be only cause in like uncomfortable for our body, but it will impair neuron proliferation in a fetus. Okay, so it should be like very careful in the first trimester of pregnancy. And then another example is low blood glucose. Yeah, if it happened before birth, it will impair the brain development as well. And as I mentioned before, the infant brain is highly vulnerable to damage by the alcohol. And many research suggests that children of mothers who drink heavily during pregnancy are born with fetal alcohol syndrome. It's a condition marked by hyperactivities, impulsiveness, difficulty maintaining attention, varying degrees of mental retardation, motor problems, heart defect, and facial abnormalities. And drinking during pregnancy leads to the thinning of the cerebral cortex, yeah, that cause this impairment. That persists to adulthood, and it's not curable. Yeah, and then more drinking causes greater deficit as well, but even moderate drinking produces a miserable effect of negative effect as well. So exposure to alcohol damages the brain, yeah, in several ways. At the earliest stage of pregnancy, it interferes with neuron proliferation as the first stage. Yeah, and little later, it impairs neuron migration and differentiation, and later it impairs synaptic transmission as well. So it's impact all of the stage of neuron development. Because alcohol kills neuron partly by apoptosis. So to prevent apoptosis, a brain neuron must receive input from the incoming axon, right? And what alcohol do is the alcohol actually inhibits the receptors for glutamate is the brain main excitatory transmitter, and enhances receptor of GABA is the main inhibitory transmitter, right? Because of the decrease in net excitation, many neurons undergo apoptosis. And further harm occurs after about drinking while the alcohol is washing out of the system. And while the alcohol was inhibiting the glutamate receptors, many neurons then compensated by quickly building more glutamate receptors. Then, when alcohol leaves the body, glutamate overexcites its receptor, bringing excess sodium and calcium into the cell and then poisoning the mitochondria. That's why the result of this alcohol intoxication is increased cell death in several brain areas. And the developing brain is highly responsive to many influences from the mother as well, yeah? Not only the alcohol, but many things. For example, if one experiment suggests that a mother of a rat is, when a mother of a rat is exposed to stressful experience, then she becomes more fearful. She spends less than the usual amount of time licking and grooming her offspring. And her offspring become permanently more fearful in a variety of situations. So analogously, the children of impoverished and abused women have, on average, increased problem in both their academic and social life. So the mechanism in humans are not the same as those in rats, but the overall principle are similar, okay? And stress to the mother changes her behavior in ways that changes her offspring behavior as well. And other substance that also influence the brain development is also cocaine and smoking that may cause a lot of things such as low weight, sudden infant death syndrome, or ADHD, intellectual deficit, and immune disorders in the infant. All right, so as we have known that neurons differ in shape and chemistry, right? They're really specific. It's when and how does neuron decide which kind of neuron is going to be? That's the question. Actually, it's not a sudden decision. So McConnell in 1992 experiment research translated immature neurons from one part of the developing cortex to another, and they found that they develop properties characteristic of their new location. However, neuron transplanted at a slightly later stage of life that develop some new properties while retaining some old ones. Okay, so we can like analogy. We can have the analogy like it's like the speech of immigrant children. So those who enter a country when very young master the correct pronunciation, whereas the older children retain an accent. In one fascinating experiment, researchers explore what would happen in the immature auditory portion of the brain if they received input from the eyes instead of the ears. I'm sorry. So the researchers conduct an experiment with a ferret. It's a mammals, yeah, in the weasel family. They are born so immature that their optic nerves from the eyes have not yet reached the thalamus. So on one side of the brain, researchers then damage the superior collicus and the occipital cortex. It's the two main targets for the optic nerves, right? And on that side, they also damage the auditory input. Therefore, the optic nerve could not attach to its usual target, and the auditory area of the thalamus lacked its usual input. As a result, the optic nerve attached to what is actually the auditory area of the thalamus. So what would you guess happened? So the question is, did the visual input cause auditory sensation, or did the auditory areas of the brain turn into visual areas? The result is actually surprising to many, and the result is what would have been the auditory thalamus and cortex is very organized. They're developing some, but not all, of the characteristic appearance of visual area. But then how do we know whether the animals treated that activity as fission? So remember in this experiment that the researchers then performed this procedure on one side of the brain, and they left the other side intact. And the researchers presented stimuli to the normal side of the brain and trained the ferret to turn one direction when they heard something and the other direction when they saw light, as you can learn from this figure of the procedure of the experiment. And after the ferrets learned this task well, then the researchers presented a light that the rewired side could see, and the result suggests that the ferrets turned the way they had been taught to turn when they saw something. And that suggests that the rewired temporal cortex receiving input from the optic nerve and produce visual responses. And then let's continue to learn the fine-tuning of our neurons by knowing that experience also take parts in tuning of our neurons. So Schatz in 1992 suggests that because of the unpredictability of life, our brains have evolved the ability to remodel themselves in response to the experience. And we continue to learn about the dendritic branching and the experience. And some decades ago, researchers thought that adult neurons substantially change their shape. Yeah. Also, the central structure of dendrites becomes stable by adolescence. The peripheral branches of dendrite remain flexible throughout life. And it can be learned from an experiment that injected a dye that let them watch the structure of a living mouse neuron over days or weeks. And they found that some dendritic branches extended between one viewing and another, whereas others retracted or disappeared. So about 6% of dendritic spines appear or disappear within a month. But then the gain or loss of spines means a turnover of synapses, which relates to learning. And experience is actually guided the neuronal chains in our brain. Yeah. So let's start with a simple example. So decades ago, it was typical for a laboratory rat to live alone in a small gray cage. Yeah. Imagine by contrast, several rats in a larger cage with a few objects to explore. And then from that differences, researchers called this an enriched environment, but it was enriched cage actually. So a rat in the more stimulating environment developed a thicker cortex, more dendritic branching, and improved learning. As a result of that research, most rats today are kept in a more enriched environment than was typical in the past. And further research found differences between the brains of laboratory real rats and wild rats that someone captured. So here, the differences is not just matter of less enriched versus more enriched, but also different types of stimulation and activity. So the wild-caught rat had more neurons in the visual areas of the brain and fewer in the auditory areas. And a stimulating environment enhances sprouting of axon and dendrites in many other species also, yeah? So we might suppose that the neuronal changes in an enriched environment depend on interesting experience and social interaction. Okay. Based on the research that has been done, the enhancements produced by the enriched environment is due to physical activity. Yeah, because in the rat cage is using a running wheel, enhances growth of axon and dendrite as well as learning, even for rats in isolation. And the question is, can we extend this result to human? So physical activity appears to be as beneficial for brain functioning in human as in the laboratory animal. So actually, this result on enriched environment is more debatable later. So remember that the studies with laboratory rats merely show that having a bigger cage with something to do was better than a small gray cage was not with nothing but food and water. So far as that goes, yes. The answer is yes. Humans in normal environment do better intellectually than children in orphanage, where the staff will provide little more than minimum care, right? But the more important question is how much we could increase intelligence beyond normal by providing special training or enhanced experience. So educators here have long operated on the assumption that training children to do something difficult will enhance their intellect in general. So for example, like training in language or calculus and etc. Yeah. The psychological term is the FOR transfer here. Near transfer occurs if training on one task produces improvement or similar tasks. So this FAR transfer is actually has a weak effect. So many studies have attempted to improve memory or intelligence by computerized tasks that practice verbal and spatial skill, for example. So actually, despite high claims by the publisher of this program, most studies show little or no improvement in reward performance actually, yeah. And typically people will show clear benefits on the skill they had practiced, especially right after the training, but little improvement of any skill, unlike the practice one, and declining benefit as time passes. Yeah, so that similarly also happened with people advise old people to do crossword puzzle or Sudoku to exercise their brain. Yeah, so actually experimental studies suggest that practicing this kind of activity improve their skill at the puzzle, but not much else. All right, and then so far, the generalized training program to enhance overall intelligence have produced only temporary or modest benefits, right? Research confirmed that. However, prolonged practice of particular activity does produce definite brain changes that enhance the ability to perform the task. So we can see that what happened to the brain if one sensory system is impaired. So we can see that people often say blind people become better than usual at touch and hearing. Well, that statement is true anyway, but we need to be more specific there. So blind people actually improve their attention to touch and sound based on practice, right? And researchers found that blind people have greater than average touch sensitivity in their finger, especially blind people who read Braille and therefore practice their finger sensitivity extensively. So just as people who were blind from early age become more sensitive to touch and sound, people who are deaf from an early age also become more responsive to touch and vision. Just as touch and sound come to activate what will be in the visual cortex in blind people, touch and vision come to activate what will be in the auditory cortex in deaf people. Okay. And then people who develop expertise in any area spend enormous amount of time practicing, generally beginning in childhood, and it seems reasonable to look for corresponding changes in the brain. And the favorite choice of researchers is to study the musician, because researchers might able to look for changes in their brain that are responsible for hearing and finger control. And secondly, that serious musicians are numerous and easy to find, and that's why it's easy to conduct research at them. One study used MEG, record the responses of the auditory cortex to pure tones, and the responses in the musician were about twice as strong as those in non-musician. And another study using MRI found that one area of the temporal cortex in the right hemisphere was about 30% larger in the musician compared to non-musician. And other studies also found enhanced response of subcortical brain structure to musical sound and speech sound compared to non-musician. So even as little as 3 years of musical training in childhood produces a measurable increase in brain stem responses to sound. And these brain changes help musicians attend to slight changes in sound that other people might not distinguish. And according to a study using MRI, gray matter over several cortical areas was thicker in professional musicians than in amateurs and thicker in amateurs than in non-musicians, as you can see in this figure. And the most strongly affected areas is related to hand control and vision, which is also important for reading the music. And a related study on stringed instrument players found that a larger than normal section in the somatosensory cortex in the right hemisphere, because it's devoted to representing the finger on the left hand, yeah, which is they use to control the string. And these results suggest that practicing a skill is reorganize the brain to maximize performance of that skill. Okay, so it's only specific to that skill. However, an alternative hypothesis is that the brain characteristics that were present from birth attract people to one occupation or another. But is it true? Let's check them. But researchers then later found that the brain differences might the result of musical training, but not the cause actually. Yeah, it's based on their research. Another issue is whether music training produces bigger effect if it begins in early life, while the brain is more easily modified. And the answer for that question is yes. Several studies found major differences between young adults who started music training in childhood and those who began as teenagers. And brain also changes after briefer practice. As you can see, as you learned before that being blind or deaf from birth will lead to altered brain anatomy, right? And so does long-term musical training. But the question is, might previous experiences also modify brain anatomy? And in a sense, the answer is actually yes, of course. Anything you learn must have some effect on your brain. Yeah? Just reading this sentence in the PowerPoint or hearing my lecture has rearranged a few molecules in your brain, and the tissue, the issue is whether a relatively brief experience produces a big enough effect that we might observe it with MRI or similar technology. Actually, that many studies have in fact reported changes in adult brain anatomy from tasks such as learning to juggle 3 balls, 16 hours of playing a complex video game, and 40 hours of playing golf for the first time, yeah? However, skeptics have raised objection because many of these studies compared mean MRIs of the entire brains of the trained group to the entire brains of a control group. So in effect, they tested a huge number of hypotheses at once, one hypothesis for each brain area. All right. And so we have discussed that playing music or practicing anything else will expand a relevant brain area, right? And the change is good actually, but not always, yeah? So brain reorganization can happen to a musician or people that play musical instrument for many hours a day for years. They represent the focal hand dystonia. Yeah, is the hand increases in the somatosensory cortex. Yeah, so imagine that the normal representation of the finger in the cortex is as follows, as you can see in the figure, but later because of their extensive training in music, playing piano or guitar, that will stimulate of one finger excite mostly the same cortical area as another finger. And it causes the musician cannot clearly feel the difference between one finger to another, and it's difficult to move them independently, right? Furthermore, the motor cortex changes also, so the representation of the middle fingers expand, overlapping and displacing representation of index finger and little finger. And similar problems sometimes also happen to writers, surgeons, golfer, or anyone else who repetitively practice precise hand movement here. All right, so as people grow older, their behavior also changes, right? But the question is how much of these changes has to do with the brain? Let's consider of two examples. The first one is the adolescents first. So adolescents are widely regarded as impulsive and prone to seek immediate pleasure based on research as they compare to adults. Impulsiveness here is a problem if it leads to a risky driving or risky drinking, risky sex, spending sprees, and so forth. So in addition to being more impulsive than older adults, children and adolescents tend to discount the future more than adults do. For example, which would you prefer? $100 or $125 a year from now? What about $100 now versus $150 a year from now? How much bigger would next year payoff have to be to make you willing to wait? So based on this example, adolescents are more likely to choose the immediate reward than adults are in a variety of situations. And many studies also have found that adolescent humans show weaker responses than adults do in the areas of the prefrontal cortex responsible for inhibiting behavior. And furthermore, the degree of maturity of the prefrontal cortex and its connection correlates positively with restraints of impulses. However, also the prefrontal cortex is indeed not quite mature in adolescent. Its immaturity is only a small part of the explanation of impulsivity. So in laboratory tests, most adolescents inhibit impulses just as well as adults. So most of the riskiest behavior, especially antisocial risky behaviors, come from individuals with a lifelong history of troublesome behaviors beginning in childhood and extending into adulthood. And furthermore, if risky impulsive behavior were the product of an immature prefrontal cortex, we should expect it to decline over the teenage years as the cortex gradually matures. But in fact, most types of risky behavior become more common toward the later teenage years. As you can see in this figure is the result of the research. And the also more likely explanation of risky adolescent behavior is that the brain responds to reward, especially anticipation of reward. It increases strongly during the teenage year. And so that the adolescents seek excitement, especially when they're trying to impress their peers. And then we also will take an example from the brain of old people. So many studies confirm that on average, old people's memory and reasoning begin to fade, and many neurons lose some of their synapses, and the remaining synapses change more slowly than before in response to experience. And the thickness of the temporal cortex shrink about 0.5% per year on average, and the frontal cortex begin thinning at the age of 30. Right? It's really brief, yeah. So the volume of the hippocampus also gradually declines in old age, and certain aspect of memory, because hippocampus is responsible for memory, will also decline in proportion to the loss of hippocampus. And old people are also decline rapidly after an injury or illnesses because of the brain inflammation. In general, old people in general who remain physically fit, they also retain their cognitive ability. Also, even those who may be slower in certain intellectual activity have developed a great base of knowledge and experience of in the older people. Also, in certain question, older people do significantly better than younger people. So it doesn't mean all is like degrading in old age. And many older people also find ways to compensate for losses of their brain function, such as by activating more widespread brain areas to compensate for decreased arousal in one or two areas. Yeah, so some procedure that might help to protect against cognitive decline in old age, including based on experimental studies in people, including the daily exercise or sedentary activities. They suggested that generally improve the cortical activities, attention, and sometimes memory. Also, chemical intervention are also worth investigating in the future, because from research in rats, old mice that get transfusion, blood transfusion from young mice, they showed a temporary impairment of synaptic plasticity and learning. And transferring blood from young mice to old ones increased the number of dendritic spines and improved their learning and memory. However, this procedure that work well with rats or mice yield disappointing result in human. It means like it's not proven yet in humans, so we need to await further research before getting too excited about this. But in the meantime, right now, if you need a blood transfusion, hope that you get it from a young person, so it will benefit you more. All right, so that's all for the topic of brain development. It's quite long explanation, and I will see you shortly in the next video. Bye.